just to recap, the first part of energy generation that we've covered is to talk about glycolysis. And glycolysis basically takes a glucose molecule, which came into the cell by facilitated transport, and in the cytoplasm, we invested 2 ATPs into a series of enzyme reactions that first made our glucose molecule symmetric, split it in half, and then it continued, those two halves continued in parallel pathways to generate some energy. We made an NADH, which we've talked about as an energy carrier. NAD plus gets reduced to NADH, and we're going to see in this podcast what happens to NADH as it moves through the procedure of cell respiration. And then, in addition to investing our 2 ATP, we finally make 4 ATP, 2 on this side, 2 on this side. And those ATPs are made via an enzyme actually connecting that phosphate to that ADP and making ATP. So it's a one-to-one. Enzyme has to do it. It's not very mechanized like we're going to see in the next steps in the electron transport chain later. And this is called substrate level phosphorylation. And here's a diagram to give you a visual of that. Basically, an enzyme is grabbing something that already has a phosphate attached to it, taking that phosphate, moving it over to ADP, and we end up releasing whatever that product is that doesn't have phosphate anymore, and ATP. So this is called substrate level phosphorylation, and that's the only way that you make ATP during glycolysis. You make a very little bit of ATP, but for organisms that survive solely on glycolysis, and we'll talk about those when we talk about fermentation, you can survive for a while. You make a little bit of ATP. You come out with 2 ATP. So at the end of glycolysis, we're still in the cytoplasm. We have 2 molecules of this pyruvate. We made 2 NADHs. We made 4 ATP. So we've got a little bit of energy. Next, we have to move into the mitochondria. And I dug out this picture from the old textbook to kind of put everything in perspective. So in the cytoplasm, we just finished talking about glycolysis. We make substrate level phosphorylation events to make ATP. We also made some NADHs. We're carrying electrons on the NADH now. Now the next steps that we're going to talk about are going to happen inside the mitochondria. So we're going to talk about the NADH gets imported in there. Actually, we're not going to talk about that. We just know what happens. And then we're going to talk about how pyruvate gets imported to the center of the mitochondria or the matrix of the mitochondria. We're going to do the citric acid cycle there. Again, we'll make a little bit of ATP. Each molecule of pyruvate that goes around this system makes 1 ATP. Again, substrate level phosphorylation, and then make a lot of NADHs that will go over to the electron transport chain and make tons of ATP. So that's the place we're going right now. So let's import our pyruvate, which is a 3-carbon molecule, and it's going to have to cross 2 membranes. Because remember, there's an outer membrane and an inner membrane in the mitochondria. We're going to end up in the matrix. This transporter complex is not just one protein. It's actually a 50 subunit complex that the books really try to gloss over. But I think we should just appreciate the significance of this molecule. It's pulling the pyruvate across 2 membranes. As it does that, it cleaves the CO2 off. So we're going to break the bond right here. Release the CO2. This is the first time that we're starting to make that stuff we exhale. We're actually making it at the cellular level, and it crosses over into the bloodstream, and we can exhale it later. Every time you break a bond, and I think I'll say this at least 50 times in this unit, we can transfer the energy to something. And the guy that's going to collect that energy as NAD+, is going to get reduced to NADH plus hydrogen. Again, we're going to use that NADH in the next step, electron transport chain, later on we'll talk about. When we break that bond, we also use the energy to attach this cofactor called coenzyme A. So coenzyme A is a organic molecule with a sulfur that's connected to our 2-carbon thing called an acetyl group. So now we have what's called acetyl-CoA. 2 of these carbons, the pink ones, come from glucose. Coenzyme A is a coenzyme because it's just going to basically shuttle this molecule into the citric acid cycle and then go and get recycled. So now we're inside the mitochondria. I'm going to skip that, sorry. And we are at acetyl-CoA getting dumped off into what we call the citric acid cycle. It's also called the Krebs cycle for Sirhan Krebs, and I will write that up here because I think he needs credit for it. So Sir Krebs was a scientist who figured out all these steps, people in his lab, organic chemist in the earlier part of the 1900s. Pretty phenomenal feat. Acetyl-CoA comes in, 2 carbons attached to this coenzyme A that's going to basically drop it off, and now it's going to meet up with a 4-carbon oxaloacetate that's at the end of the cycle. It's a cycle, so it's going to keep going. That end product now joins up with the new coming carbons to make a 6-carbon thing. 4 carbons plus 2 carbons equals 6 carbons. It makes something called citrate, which is actually citric acid. It's the stuff that makes your lemons tart and your oranges tart. And now we're going to take this through multiple steps. Every time we break a bond, so you're going to go from 6 carbons to 5 carbons in this step, we're going to transfer that energy to NADH, release the CO2. You go from 5 carbons to 4 carbons. Again, you're breaking off a CO2. We're going to make another bit of energy transferred over to NADH. This is a rearrangement step from 4 carbons to 4 carbons. That rearrangement step, again, we're moving things around. We're doing what's called substrate level phosphorylation again to make a GTP. That GTP also transfers its phosphate over to ATP, so that's where you're making the ATP. We're still at 4 carbons. Notice we're at 4 carbons. We're at the bottom of the circle. We have to get up to 4 carbons up here. What we're not showing you in this simplified version, again, go to biochemistry in college and you'll have to probably memorize all these like I did once upon a time. And we're basically rearranging bonds to go from these 4 carbons up to this 4-carbon structure and making basically sucking off a little bit of energy as we go along. This next step where we rearrange some bonds, we're making this new one called FADH2. It's sort of like a cousin to NADH. It's worth a little bit less energy. I'll show you that when we get to the electron transport chain, but it's an energy carrier nonetheless. Again, rearrange a few more bonds, make one more NADH, and now we are back to that starting material, oxaloacetate. I like this diagram for its simplicity. Don't worry about the names of the intermediates. What things you want to worry about are the names that are here. Acetyl-CoA is a 2-carbon thing that enters a citric acid cycle. It binds a 4-carbon thing called oxaloacetate to equal a 6-carbon thing called citrate acid, actually. We're going to break bonds. Every time you break a bond, you make an NADH, so we lose 2 carbons in CO2. We make a GTP down here. We rearrange bonds and capture the energy in an FADH2 and an NADH2. So this acetyl-CoA is half of a glucose molecule. So for every glucose molecule, this goes around twice. So if we do some accounting, we make 1, 2, 3 NADHs times 2, so that'd be 6, 2 FADH2s. So we're starting to capture a lot of energy that we need to go drop off at the electron transport chain. And here's the electron transport chain. So again, this is a picture from the previous edition of the book. We are inside the mitochondria. So right in here is the matrix, and this is where we've been doing the Krebs cycle, or the citric acid cycle. And this orange area is the inner membrane space. I'm sorry, I paused because I was looking for a picture of mitochondria, which you see here. So the center here, yellow, is the matrix. That orangey part here would be the space between the outer membrane and those folds, and that would be the inner membrane space. So the electron transport chain is basically a series of proteins and a lipid that are embedded or associated with the inner membrane or the cristae, and the electrons are going to be passed off, so we are going to reduce these in order, the chain, and ultimately build up a concentration gradient of hydrogens up here. Remember, gradients can do work for us. They have potential energy. We're going to use energy to make the gradient, and then we're going to allow that gradient to go drive this enzyme called ATP synthase to make ATP. I think this video should be done in 2 parts, so I'm going to stop here and then pick up again with the electron transport chain so as not to overwhelm you.